This is really a great honor and privilege, again, like all of our speakers, for me to introduce Dr. Diane Chugani. And I hope I'm pronouncing your name right. Is that okay? I'm assuming that's Italian. Is that correct?
I'm Italian, but that name is Indian. It's my husband.
Oh, well, so much for my cultural sensitivity there. So Dr. Chugani is Chief of the Division of Clinical Pharmacology and Toxicology, as well as Director of the Translational Imaging Laboratory at the Children's Hospital of Michigan, which is in Detroit. She is a professor of pediatrics and radiology. She founded the Children's Hospital Autism Center in 2008. The aim of Dr. Chugani's research is to discover mechanisms involved in the pathogenesis of autism and to design and test new treatments based upon research findings. Sounds pretty relevant, doesn't it? Her studies of serotonin synthesis in children with autism have identified differences in developmental changes in brain serotonin synthesis in young autistic compared to non-autistic children. Based upon these results, Dr. Chugani conducted a pharmacokinetic safety and efficacy trial of a serotonergic drug in young autistic children, which is funded by the National Institute of Child and Health Development through the Pediatric Pharmacology Research Unit Network. And based upon the preliminary findings of that study, Dr. Chugani was recently awarded an Autism Centered Excellence Network grant to further test this novel treatment. And so I'm sure she can tell you a little bit more about how we first connected, but I'll just say that we are truly delighted and honored and privileged to have you here today, so thank you. Don't forget your wine. Mm-hmm. Don't forget your wine.
My wine.
Yeah.
I can't forget my wine.
No. You set? You got everything you need?
We'll find out.
Okay.
Okay, well, I'm very thankful that Paul called and emailed me so many times to come to this meeting. I first heard about Jacobsen Syndrome about a year ago. Paul, when did you first call me? Do you remember?
I've called so many women, I don't know.
Anyway, you know, it was a regular day, and it's always nice to have an interesting surprise. So I answered the phone, and, you know, Paul said that he had seen a child and had talked to a mom at a meeting who said that her child had participated in my autism study, and the mom thought that the child had done very well, and he wanted to know, you know, whether I knew whether the child had gotten the drug or not. And, you know, what Dr. Stein said this morning was, you really, in order to test drugs, you need to do a placebo-controlled trial. So at the time he first called me, we didn't know whether this child had gotten the drug or not. And when he called me, I went back and looked at my source notebook to see what kind of data we had collected. And although the testing showed that she really hadn't improved on her autism, she had dramatically improved on the scale that Natasha talked about this morning, the Vineland Adaptive Behavior Scales, a very dramatic improvement over 6 months that really wouldn't normally be expected. So I was pretty excited too. But of course we didn't know. So we actually had to wait until this year to, when the study was unblinded, to know whether she was on the drug or not, and she was in fact on the drug. So we, this study is still under analysis. We don't know the results of the whole study, and I'm really not at liberty to talk about any of the analyses that we've done yet. Having said that, and I would also like to say that the result in one child doesn't mean that every child is going to have that result. However, due to your greatest advocate, Paul Grosveld, and many phone calls and emails, I decided to come. The reason I decided to come is because I think there is potential to, you know, to help other children. So I'm just going to put out there what we've done, and then I kind of look forward to hearing the discussion with the group. Okay. And I think Paul gave this title too. I'm not sure. Did I give you this title?
Let's see, did I come up with that?
I'm not sure.
No.
I came up with that? Oh, gosh.
Because I wouldn't have been that specific.
Okay.
By the way, I think you were the first one to call me. I emailed you and you called me back. So let's set the record straight.
It must have been a very intriguing email because a lot of times I don't call people back.
No, it's true.
Okay, I don't have that kind of memory. So, the speakers this morning really set this up nicely for me, and I'm just going to kind of repeat a few things that I think are important to kind of tell the story. So, again, I think as I think Natasha said this morning, we used to think about autism as being a rather homogeneous group, and that there were a few disorders like tuberous sclerosis or Fragile X or Rett syndrome in which maybe half or a large fraction of those kids might have autism, and then for all of the rest of them that we didn't know what they had, we thought it was homogeneous. And when we wrote studies to do imaging studies or to do a drug trial, we really had to exclude those kids that had Fragile X and tuberous sclerosis and Rett syndrome, and then we took all of the rest of the comers. That's changed now, but that's how our study was done. So we were very interested in serotonin and autism, and I'm going to tell you that serotonin gets affected in many of these different causes of autism. So I'm going to kind of argue that serotonin is a target for many different kinds of etiologies. So what is serotonin? Serotonin is a chemical that's in our body. Can I do pointing with this?
Yeah.
Okay. So this is kind of a cross-section of the brain like this, and through the midline, and right here you can see this big red spot. This is where all of the cells in the brain that are making serotonin are located, but they send processes all over the brain. So actually serotonin and the enzymes that are making serotonin are found throughout the brain. They're found in the cerebellum and the thalamus and the cortex, all throughout the brain. And serotonin is also made all throughout the rest of the body, and this is a slide from a review article. So here we have the brain, we have the GI tract, and actually if you look at the literature, a lot of people will say that about 5% of the serotonin is made in the brain, and then the rest of it is made in the GI tract. Well, in fact, serotonin is made all over the body, and actually this doesn't show it, but serotonin is made in our skin, it's made in our lungs, it's made in our blood vessels. And I really was interested in hearing the talks about the platelets. The platelets don't make serotonin, but they take up serotonin, and serotonin is very important for their function. So one of the things that I thought about a lot after Paul contacted me and I read a little bit about Jacobsen Syndrome is what might be happening in the platelets as well as autism. So one of the very first findings, chemical findings about autism, is that there's elevated serotonin in the platelets of up to half of people with autism. That was first reported in the literature in 1961. It's been replicated many more times. I could put a lot more articles on here. So we know that in a large portion of people, even though like maybe 1% has this gene and 1% has this gene, 1% has this gene, up to 50% have elevated serotonin in the blood. There's also elevated serotonin in a lot of their relatives. So people started trying to find out, well, what's going on with serotonin in the platelet. And serotonin, there's something called the serotonin transporter. There's a serotonin transporter on your platelets. There are also serotonin transporters in your brain. So serotonin is on the transporters on the cell membrane, and it takes up serotonin into the cell that it's on. And so some genetic studies showed that there was an interaction between genetic variation in the serotonin transporter and another gene called an integrin gene that interact to take up serotonin into the platelet, so that had been shown. And there's a mouse model where there was a mutation in the serotonin gene from humans. It was made into rats, and those mice, and those mice also showed high serotonin in their blood. So at least one thing that can regulate serotonin in the platelet is the serotonin transporter. So people have looked for a long time to see, is the serotonin transporter, is that the answer to autism? Well, it's the answer to autism for a small percent of people. So people have reported polymorphisms of the transporter, promoter region of the serotonin transporter gene to be associated with autism. Again, that interaction between the serotonin transporter and this beta integrin. And some of the variants in the transporter were associated with rigid compulsive traits in people with autism. There have been studies of serotonin receptors in autism. So a study by Declan Murphy in UK of adults with Asperger's syndrome, which is, we call that high-functioning autism now, showed decreased cortical receptor called the serotonin 5-HT2A receptor. Another study showed lower 5-HT2 receptor in the parents of children with autism using PET. And so if you think of some of the other genes that are associated with autism, one of them is Fragile X. So there's altered serotonin synthesis in a fruit fly model of Fragile X. There's also altered serotonin systems in Rett syndrome, another genetic disorder in which autism is associated. So there are decreased levels of the breakdown product of serotonin, called 5-HIAA, in the cerebral spinal fluid of patients with Rett syndrome. And then, again, there are these mouse models of Rett syndrome, and they show some differences in serotonin concentrations in the brain, and those change over age. And also, it has been shown that treating with the SSRI, like the doctor talked about this morning, fluoxetine actually regulated the transcript, the gene for Rett syndrome. And there are also some non-genetic associations with autism. Some of those have to do with maternal infection. And there are a number of animal models of maternal infection or inflammation. In a maternal influenza virus model, there's decreased serotonin and decreased metabolite in the cerebellum of the brain from Hussein Fatimi's group. And Sujatha Kannan, who started working with me, and now she's at Johns Hopkins, has a model of maternal inflammation using lipopolysaccharide, which is a bacterial product. And when she injected that into the uterus of some rabbits, she saw a decreased serotonin postnatally in those rabbit kids. And I told you a little bit about these transgenic mice with the mutation that was from a child with autism. And these mice not only have high serotonin in the blood, they have enhanced serotonin clearance from the brain, but a normal serotonin concentration. They have altered sensitivity to receptors, the 5-HT1A receptor and the 5-HT2A receptor. The 5-HT1A receptor, I want to remember that because buspirone, the drug that I'm going to talk about, is a partial agonist. That means it acts like serotonin partially at this particular receptor. These animals were put through some of the marble burying that we saw this morning and other social tests, and so the repetitive behavior was the marble burying. For social function, instead of smelling the Q-tip, in this case, they used a three-chamber model in which there was another animal in one chamber, and the one they were testing was put in the middle, and so if they're social, they'll go to the other animal. If they're not social, they'll go to the other side. So again, sort of an interesting model about how serotonin might be affected differentially in the brain and in the platelet. So what about Jacobsen syndrome? So when I started reading about it, I got pretty excited that there were platelet abnormalities that might be related, or at least give us some information about what might be happening in the platelet and autism. So we heard a lot about platelets yesterday, but the doctor didn't really specifically talk about serotonin. So I think this is a study from your group, Paul, the white paper.
No, that's actually,
No?
he's in, I want to say South Dakota, maybe?
Okay, so they showed that the platelets have reduced numbers of serotonin-rich dense bodies. I think she did show us yesterday that there are less of these dense bodies in the platelet, if you can remember from yesterday, and also that there's this abnormal fusion of the alpha granules in the platelets. So how might these 11q deletions affect platelet serotonin and brain function? So in my conversations with Paul over the past year or so, we did quite a bit of talking about neurogranin, and he told me about RICs, and I, like any self-respecting scientist, I went and read about all those things. And so I think that there are some kind of interesting things about it, which I'll go to in a minute, but just to kind of reiterate that neurogranin is a very abundant protein in the brain, as Paul mentioned this morning, but it is also very abundant in the platelet. And so neurogranin binds with another protein called calmodulin, and in the synapse, they regulate a process called long-term potentiation through binding with calcium. So part of learning and memory involves this process, and I know you guys were kind of wondering, well, what does neurogranin have to do with memory? So I'm going to try to talk about that a little bit. And then the other one he mentioned this morning, this RICs, which is a Rho GTPase activating protein, and it binds to other proteins called these Rho GTPases and regulates their function. So we knew that neurogranin was high in the brain, but I was wondering, is neurogranin expressed in platelets? And in fact, it is the second highest expressed gene in platelets, and this is a paper here where the first highest expressed one is here, and the second one is the neurogranin gene. So what might this have to do with platelet function, this neurogranin and these Rho GTPases? So I actually put the whole figure legend here because it's kind of confusing even for me. So I'm going to kind of walk through this and then kind of show you what's happening over here. So the first thing is that platelets, in addition to having the serotonin transporter, that's this right here where the serotonin is taken up into the platelet from the blood, it also has right here this 5-HT2A receptor, so it's a serotonin receptor. So serotonin in the blood can interact with the platelet in two ways. It can be pumped into the cell, or it can bind here and start a series of reactions happening. Okay, so upon stimulation of platelets by serotonin, the phosphatidylinositol pathway is activated, resulting in a rise of cytoplasmic, that means inside the cell, calcium. Serotonin, let's see, the serotonin signaling is mediated by the G protein 5-HT2 receptor, I already told you that, and by import into the cell of, by SERT. Okay. So then calcium, so you've got your calcium. So I was telling you about how, so neurogranin binds to calmodulin, calmodulin binds up the calcium. So we've got, so the CAM here is the calmodulin, and we've got the calcium being activated in the cell. And although it's not on here, we know that from this other paper that neurogranin is expressed in the platelet, so somehow it's regulating how much of the calcium calmodulin is available for this process. Okay, so then, where are we? Okay, so then the calcium activates another enzyme called transglutaminase. So here's the transglutaminase, and the transglutaminase takes the serotonin that's been pumped in through the serotonin transporter into the cell, and it hooks it on to small GTPases, these Rho GTPases. Remember we mentioned that RIX is a Rho GTPase activating protein. Let's see, where am I now? Oh, I'm all lost. Oh, here we go. Okay. Okay, so the transglutaminase hooks serotonin onto Rho A and also to RAB-B, which is also a Rho GTPase that's associated with the alpha granule, and blocks the GTP hydrolyzing activity of these proteins. Okay, so then calcium in a complex with calmodulin activates guanine nucleotide exchange factors. So that activating protein, the Rho activating protein, or RIX, is one of those types of proteins. Not clear whether it's in the platelet or not, but maybe it is. It's in the brain. And causes to change the function of this Rho A and RAB-B. I think I'm not going to read any more, but just to kind of give you a feeling about how all these players that we're talking about, neurogranin, Rho GTPase activating proteins, these are all involved in the regulation of the storage of serotonin and the release of granules from the platelet, and these processes may be part of the changes in activity in the platelet. Okay, so what about the synapse? So all these players are in the synapse too. So in a way, we might be able to think about the platelet in Jacobsen Syndrome as sort of a model for what might be happening in the brain with these very same proteins. So the neurogranin is known to be highly enriched in the dendrites, and the dendrites, so the way that nerve cells communicate with each other is that one cell has its dendrite and it's got neurotransmitter in it, and then it releases neurotransmitter which binds to receptor on the next cell. But then there's all kinds of other signaling that happens as well, and as there's activity between two cells, either that synapse gets strengthened or not. And some of these proteins are what's involved in the growth of the synapse to make it stronger, or the retraction of the synapse. So all the same players are here. So neurogranin is here, the calmodulin, the calcium calmodulin is here, Rho A is here, RAC1 is another Rho GTPase, and all of these are involved in the strengthening of the synapse, or the long-term potentiation that happens when these cells get activated. So the cells get activated by glutamate, they bind at this NMDA receptor, and then there are changes that happen in all of these pathways. Now, people were sort of asking me questions about, well, you know, why would somebody have a mutation in that it includes neurogranin, but different from another child? Well, if you can imagine, you can kind of mess this up by having changes in many of these different proteins. And in fact, many of the different proteins involved in synaptogenesis, and then even more that are involved with the connection between the two neurexins and so on, in the formation of the synapse, have been identified as those genes that are associated with autism. So although we say that there's 400 different genes, it's not like 400 random genes. A lot of the genes are involved in the function and the formation of synapses. So that's kind of one big class of proteins that have been identified that are associated with the risk for autism. I'm just going to take a little drink. I hope I'm not putting everybody to sleep after all that pizza. Okay, so and this, so the last slide with the platelet, it was pretty obvious where the serotonin was involved. In this case, the slide doesn't actually show it. I couldn't find something that had calmodulin and Rho A and RAC and everything, but it's known that particularly in the hippocampus, there's a high concentration of 5-HT1A, that serotonin receptor that buspirone acts at, and in the hippocampus, serotonin has an interaction and regulates the activity of the NMDA receptor. So that's the connection there that I'm thinking about. Okay. And another thing that's kind of interesting is that the 5-HT1A receptor has 2 binding sites for calmodulin that regulates its function. So this is a model of the serotonin receptor. There's a whole bunch of different receptors that have a kind of we call it a 7-transmembrane type of receptor, and they interact with G proteins, and they have this big loop here. We call it the cytoplasmic loop. It's inside the cell, and this cytoplasmic loop is different for lots of different receptors, and they have different regulatory areas, and right here are binding sites for calmodulin. So the 5-HT1A receptor is affected by calmodulin, and if you remember, calmodulin is the binding protein for neurogranin. So that's kind of another connection. Okay, so what did we actually do with imaging and the treatment study? I'll kind of talk about that now. So kind of going back now 10 years when we actually designed this study, we were really struck by the fact that well, at that time, really, we didn't even know about all these genes. But we did know that serotonin was affected in a large number of people with autism, at least in the platelet. And studies were kind of, although I told you one where in Rett syndrome, there was a change in the metabolite and cerebral spinal fluid. Many studies have been done with other kids with autism, and there really were not very conclusive. And so what we did is we we call it molecular imaging now. We used to call it PET scanning. We labeled a tracer called alpha-methyltryptophan, we call it AMT, with positron emission tomography, and we used it to measure serotonin synthesis in children with autism spectrum disorder. ASD means something different to your cardiologist, right? Atrial septal defect, is that what it is? Yeah, okay. I saw it on the slide yesterday. I was thinking, what the heck, why does he have ASD on there? So, okay, so.
It's an easier fix, ASD.
Okay, so alpha-methyltryptophan is a modification of tryptophan. Does everybody know what tryptophan is? No. Okay, tryptophan is an amino acid, so all our proteins are made up of building blocks called amino acids, and one of them is tryptophan. So when we eat, our body digests the protein, and those amino acids are taken up into our blood, and then our body uses them for different things. It uses to make more protein, but it also uses them for other things. And tryptophan is actually a precursor for serotonin. So the tryptophan that that's taken into our bodies goes into different pathways. And, I'll just go back for a minute. So the alpha-methyl allows us to put this carbon-11, which is a positron emitter, and the way that PET works is that we take this radio-labeled compound and we inject it into the individual, and then that tracer distributes throughout the body the way that tryptophan would. And so this is actually a model that was developed to measure serotonin synthesis by Mirko Dixik in Montreal. And so the thing that's kind of nice about this alpha-methyltryptophan is that it's not incorporated into protein, so this alpha-methyl group doesn't allow it to be made into protein, but Mirko showed that it was converted in animal studies into alpha-methylserotonin, and that the alpha-methylserotonin was localized to serotonergic neurons and their terminals. And also what's nice is that the alpha-methylserotonin didn't get broken down by the enzyme monoamine oxidase, so it kind of gets trapped in the tissue, and so we use kind of a model to say that the way that this tracer is trapped in the blood is indicative of how much serotonin is being made. And then we call that the serotonin synthesis capacity, because it's not really the rate, and we use a term called the K complex to describe how we calculate it. Now, when we studied this tracer in children with autism and those that didn't have autism. And most of the children who did not have autism were being studied for their epilepsy, but they were developing normally. So, you know, they had a normal IQ and they did not have autism. And so what we saw when we plotted this according to age, so here we have 2.5 and 5 and 10 and 15 years old, that the kids who didn't have autism had higher values when they were young. So when they were, 2 or 3, they had high values, and by around the age of 5 or 6, their values started coming down. And these values here in the 15-year-old range are very similar to what we measure in healthy adults. And then when we plotted out the same thing for the children with autism, we found that their values were lower at the younger ages, and if anything, they were increasing with age, so that the regulation of the way the serotonin was being made was different in the kids with autism. And in a subgroup of the kids, we also saw some particular areas of the brain that showed differences. And so these images that I'm showing here are slices of the brain like this. So the top one is here, so higher up is here, and a little bit lower down is here, and a little bit lower down is here. And so what we saw is that there were unilateral decreases in the cortex, and the extent of that differed among the kids. And on the same side that we saw this decrease, we saw a decrease in a deep area of the brain called the thalamus. And then we also saw an area on the other side of the brain, in an area of the brain called the cerebellum, and this is an area of the cerebellum called the dentate nucleus. We saw an area of increase. And the way that we interpreted this is to kind of think about, how are these areas of the brain related? And so we thought that this may be showing us something particularly happening in what's called the Dentato-Thalamo-Cortical pathway. So one thing that has been found in autism for many years, so one of the oldest findings, like I told you, was increased serotonin in the blood. And another very old finding is like maybe from, 20 years later, 1984, Margaret Bauman and Tom Kemper showed that there were decreased numbers of a kind of cell in the cerebellum called Purkinje cells. And again, just like the serotonin synthesis, this decrease in Purkinje cells has really been replicated over a large number of studies, so we really believe that. So the Purkinje cells of the cerebellum then project to these deep cerebellar nuclei, like the dentate nucleus. Then the cells of the dentate nucleus actually project to the other side of the brain, to the thalamus, and then the thalamus projects through different parts of the cortex. So how can we think about this? How can we relate this to what we know about the brain in animal studies? And so we were very interested. There's a large number of studies looking at serotonin and the role of serotonin in the development of the cortex. And this is a slide from a paper from Patricia Gaspar showing that during development, the cells in the thalamus that project to the sensory cortex, these cells transiently express the serotonin transporter. And interestingly, these are glutamatergic cells, so they're not making serotonin, but they're taking up serotonin that's being released in the cortex. And this is kind of showing some different things, but I'm going to talk about it in quite a little bit different way. So this comes from a part of the brain called the barrel field. And the barrel field actually represents, in the rodent, the whiskers that they have. And so each whisker is sort of represented by a distinct territory in this cortex. And in the wild type, that is no change to serotonin, they have a particular orientation. Now, here they're showing an MAOA knockout. So remember, MAOA is the enzyme that breaks down serotonin. So in the MAOA knockout, they have 7 times too much serotonin in their brain. And then if you look at how these cells project, they don't obey the barriers here. And so if you touch one whisker, it should be discrete, but when there's too much serotonin, it's not discrete, so it's changed their sensory function. And I thought that was really interesting in terms of autism, because if you think about it, probably their interpretation of sensory function is altered compared to us because they're not really experiencing things the same way that we are. So, taking these things together, this difference in serotonin synthesis with age, understanding that serotonin is important in brain development. We did a pilot study a couple of years ago. This was the one that Paul mentioned from the Pediatric Pharmacology Network. So we thought, well, if serotonin synthesis is less in autistic children who are between the ages of 2 and 6, serotonin is important for postnatal synaptogenesis and actually neurogenesis as well. We thought that one way to treat autism might be to give a serotonin agonist. And really the goal here was not like we were talking about this morning, not to target aggression or hyperactivity or whatever, but rather to target an abnormality in the chemical process, sort of like if you are diabetic and don't have enough insulin, you give more insulin, sort of that kind of a concept. And for our study, like I said, we chose this 5-HT1A serotonin agonist buspirone. We had a lot of reasons to pick that based upon basic studies, which I'm not going to go into right at this moment. Well, when Paul heard about the doses that we used, he said, can that really make any difference? Well, actually, before we did the study, we did some pharmacokinetic analysis. We actually brought a few children to our research center for the whole day, and we gave them one oral dose, and we took blood over time. So here's So here's where they took the dose, and then we measured up to 8 hours, and we looked for buspirone itself, and we looked for it's the metabolite, we call it 1-PPP, and then we compared those blood levels to blood levels that have been done in clinical trials of other of older children and adults. And we used the doses that we used because they gave us blood levels of these of the drug and metabolites in the same range of the doses that are used clinically in older children. In addition to the rationale that I just told you about serotonin, another thing that's pretty interesting about buspirone is that it causes a release of oxytocin in the blood. And oxytocin, and this is from Larry Young's lab. So you may have heard about clinical trials what are some of the clinical trials that are being done right now? There are actually a few really big clinical trials being done right now with oxytocin. And oxytocin is a hormone, and I couldn't quite tell whether you guys have looked at oxytocin in kids with Jacobsen's, but since some of the other hormones are decreased, it may be that they have a decrease in oxytocin. There have been some studies showing in autism in general that there are decreased blood levels of oxytocin, and oxytocin is a hormone that gets released to deliver your child. It gets released when you're breastfeeding, and it also is released during sex. It's a hormone that's involved in social interaction. So there's a lot of interest in treating with oxytocin. Oxytocin, you can't take it orally. They're inhaling it. There's a little bit of controversy about when you inhale it, does it get to the brain? But there are a lot of studies happening like that now. And so this is a study in, I can't remember if this is mice or the prairie voles, so Larry studies the prairie voles, but anyway, so he did some studies looking at, are there any small molecule drugs that we can give that can release oxytocin? And here is the plasma level of oxytocin, and here's time, and here is the administration of buspirone, and you can see this is without the buspirone, the oxytocin level, and here is it goes way up with the drug. And then he also did, again, some of these behavioral tests in the animals. And so he gave an injection of buspirone, and then he did a social learning study, and then he did what he called a partner preference test. So what he actually measured is the time they spent huddling together, either the mice or the voles, I can't remember. Okay, so what he found is there was a dose-dependent effect. So if he gave nothing, these are the times they spent with either a stranger or a partner. And here is with 8 milligrams of buspirone, they spent a lot more time huddling with their partner and less with the stranger. And here at 30 milligrams, the effect was gone. And I forgot to mention when I was showing you the other slide, too much serotonin or too little serotonin is bad. So you kind of need to pick the right dose. So in this case, there was kind of a sweet spot for what would happen. And if you gave too much or too high of a dose, you would actually have less of an effect. Okay. So the big study that I'm doing, I can't talk about it all. This is the results of our pilot data. And in that study, the pilot study after the pharmacokinetic study that we did, we studied 16 children, 7 between the ages of 2 and 4, and 9 between 4 and 6. We had more boys than girls. Here are some values for some of the tests that you're hearing about this morning, the Vineland Adaptive Scale. And in that study, what we did is we did a PET scan first because we thought that kids who had very low values for serotonin synthesis would respond better, that they would need more serotonin tone. And in the study, we first, in this particular study, it was a crossover design. Half of the kids were in the placebo group and half in the buspirone group, and after 3 months they crossed over, and the kids who had the buspirone got placebo, and so on. And we did a whole battery of tests to kind of see whether there was some efficacy of the drug. You heard about the Autism diagnostic observation schedule this morning. So that was one of the tests that we did to observe what the children did at the beginning of the study and at the end. We also used a test called the LIDER, and we used the Vineland Adaptive Behavior Scales. And then we also had a bunch of parent tests. In order to be in the study, they had to meet criteria for both the ADOS and the Autism Diagnostic Interview. We also had other measures of social behavior, the Social Reciprocity Scale, and Repetitive Behavior Scale, and so on. And then in this pilot study, what we found was there was improvement in some of the autism symptoms that we talked about this morning. So social interaction was improved in the 2 to 4-year-olds, as both rated by the parents and by the examiners. And the repetitive behavior was rated as improved by, actually I think that's 1.7, by the parents. And here again is, here's the examiner-rated social interaction. Again, you could see the effect was bigger in the younger kids than the older kids. And we also looked at some associated symptoms. So the parent-reported sensory problems improved in the younger group. Sensory seeking improved across both age groups. And anxiety also improved across both groups. Now, buspirone is used in older children and adults with anxiety, so we talked about anxiety a bit this morning, as well as being a problem for some of the kids with Jacobsen Syndrome, so this is a drug that is approved for use with anxiety now. So, based upon this, we felt that buspirone was associated with improvements in 2 of 3 of those symptoms, as well as sensory problems and anxiety in that pilot group, and the gains were greater for the younger children. As we had hypothesized in this pilot study, the kids that had lower values of that K complex on serotonin synthesis did better than the kids that didn't. So then, based upon that pilot data, we were awarded a grant to do a bigger study, and this is the study that the child with Jacobsen Syndrome participated in. This is a study that actually was not only carried out at our site at Wayne State, but also at Cleveland Clinic, New York University, Rainbow Baby and Children's at Case Western, University of California, Davis, and University of Texas Southwestern. And I'm really thankful to all of the MD collaborators who worked with us on this study. So here was the awkward part. I really am not supposed to be talking about all this until the study is completed. So I'm going to stop right here with that and just mention one other thing. So remember this morning Paul mentioned about Alzheimer's and neurogranin. Well, neurogranin is also decreased in traumatic brain injury. So we know with traumatic brain injury, those dendrites get sheared. You probably been hearing about the NFL and all of that, and they have a decrease in neurogranin. And so I was kind of interested in looking to see if there were other neurogranin-related things to 5-HT1A and buspirone. And so I found a few studies in which, in animal studies, they were studying traumatic brain injury, and so there so here's one of them. So traumatic brain injury-induced loss of dendrites and cognitive deficits were decreased by chronic treatment with buspirone. So I thought that was pretty interesting, and here's some of the data. And I was really it's kind of uncanny that you were actually talking about environmental enrichment this morning as well. So I'm a pharmacologist, but I'm also a big proponent of behavioral intervention, and that's why I started the Autism Center at Children's Hospital, where we do assessment and applied behavior analysis intervention for kids with autism. So this is kind of interesting because there seems to be a combined effect of environmental enrichment and treatment with buspirone. So here in this axis we have the morphology of these neurons are in the hippocampus, and so here is, I don't know why they did it this way, they did it backwards in my opinion. So here are the intact neurons from the sham control, so they did something that seemed like brain injury, but it wasn't. And this is 100%. And then here is, I think this is vehicle. Vehicle means they just gave whatever they dissolved the buspirone in, so there was, big cell loss. It's down to about 40%. Here is with buspirone, so it went up somewhat, they increased, and here is the environmental enrichment, so you can see the environmental enrichment did a lot, actually the buspirone and environmental enrichment actually look pretty much the same here. And here it is for the behavior, and this was I think this is called a water maze, so the animals get exposed to where a platform is, and then they learn that, and then they have to come back and find the platform, so the longer time they spend looking in the right place for the platform shows how well they are doing. So again, this is the control. This is with just giving what the drug is dissolved in. Here's what happens with the buspirone, so it's not as good as, control, but it's almost there. And here is the environmental enrichment. So this kind of gives you a feel for what buspirone and environmental enrichment can do in this kind of model of traumatic brain injury. So, is buspirone a potential treatment for Jacobsen syndrome? I guess that's why you asked me to come. So I do want to reiterate that results in one child with Jacobsen syndrome may not generalize to other people with Jacobsen syndrome. and also remind you that the child in our study did not improve on autism measures, but did show quite a remarkable improvement on adaptive behavior scales. So actually, in my opinion, that's pretty exciting, because it might mean that in Jacobsen syndrome, there's a potential treatment not just for people with autism, but also those who have developmental delay, whether they have autism or not. Whoops. How do I go back? I can't stop like that without thanking all of the people that did all the work. Okay, so obviously, I don't do all of this by myself. I already acknowledged the people from the other sites who participated in the study. Here are the people at our own imaging center, the psychologist who assessed the kids, the people who did the pharmacology and recruited the patients, and PharmD who helped with pharmacokinetics, our statistician. I have to thank the NIH for their support of our studies, and also the very first study that I did was receiving a pilot fund of $25,000 from our local Mental Illness Research Association in Detroit, so I thank them very much, and I especially thank the families and children who have participated in our studies, because these drug trials are a long haul. They have to take blood and do all kinds of things, so I really appreciate everything that the families do to participate. That's it.
I'm going to guess there are going to be a few questions.
Thank you so much for coming. You mentioned that the results were more significant for the younger children. Mm-hmm. Is that something you're looking more into in this the study that you're doing? Like, would a slightly higher dose maybe make an impact? Or my son is 8.
So we don't have the answer, obviously, from the larger study, but of course, that's one of the things that we're looking at. And just like we mentioned for the ABA and early enrichment, we think that the earlier you start, the better chance you have to make more gains. But we don't know if giving the drug at a later age may or may not be beneficial.
Thank you.
Mm-hmm.
Yeah, thank you for coming. You had mentioned about serotonin and the platelets.
Mm-hmm.
From a hematology standpoint, we know the platelets don't function. Does that mean, I'm not sure if the serotonin and the platelets are used as like a transport, or if from a hematology standpoint, if they don't function, do they not function in the case of the serotonin?
I don't quite understand the question, but I'm going to try and answer anyway. So serotonin is part of the function of the platelet. So when the platelet goes to make a clot or do some of the functions that it does, it releases serotonin. So serotonin is part of that function. And the reason that I showed you all of that stuff was I was kind of trying to get across what some of the players might be and how a deletion of neurogranin or the ROGTPase activating protein and how they might work together to cause even more so there's one thing that was discussed yesterday is why do some people with Jacobsen syndrome have this bleeding problem and one not? If some of them have the deletion of neurogranin and some of them have the deletion of the RIX, if both of them are involved with that process, that would be different ways of potentially, I don't know if you buy that or not. And then the other reason I sort of wanted to talk about that is that those processes that are happening in the platelet, similar kinds of processes are happening in the brain, and we don't really have we have access to animal models. So one thing I was wondering when you were talking this morning was, is there any platelet dysfunction in the neurogranin knockout animals?
Not that I'm aware of, but I don't know if anyone has actually looked at that. I did not know until today about neurogranin and platelet function, so I think that's definitely worth looking into.
And so I don't have any connection with what buspirone might do to the platelet. I don't think that it has a role there because the action at the platelet was, you know, the 5-HT2 receptor, not the 1A. But the reason I showed that is how, like I said, how that might be a model for what might be happening in brain as well.
We're getting that feedback from us, terrible.
I was just curious, right now, can you hear me?
I can hear you, but
Right now, I did a study for ages 2 to 6, and the younger kids seem to get more benefit from the pilot study. If or when medical advancements allow you to identify that a child would have autism or another genetic, you know, if the technology becomes available, how young would you ever give buspirone to a child? From birth, if you knew that they had an issue, or how young would you do that?
I guess I would want to know that serotonin was abnormal in the brain, but yeah, I mean, I would be interested in examining that and doing it as soon as possible. Actually, there are some studies, even in utero, showing the importance of serotonin in brain development. There's so a kind of really interesting study from Alex Bonin at USC showed that actually during the very first part of pregnancy, in animals at least, that the serotonin in the fetus in the forebrain comes from the mom. It gets transported from the mom because there's serotonin in the brain before the brain actually has the enzymes to make the serotonin. So that's pretty interesting because also in the placenta is another group of enzymes that metabolizes tryptophan by another pathway called the kynurenine pathway. And that pathway is really important for preventing the baby from being rejected by the mother. It inhibits, T cells at the placenta by actually depleting tryptophan. But if the mom has an infection, that gets cranked way up. And so actually one of the things that Alex is studying right now is this playing a role in the risk of autism in moms that have infections during pregnancy, for example. So it goes all the way to the beginning.
Okay, so I think what we can do is take about a 5-minute break to set up so we can start our panel discussion. One thing I'd just like to say is an amazing, I think, tribute to how this whole group works. You know, looking back historically, I first learned about one child with Jacobsen syndrome having autism in 2004. That was Robin Olney, who I just saw again a couple months ago in Germany. And I think I told you at that point I was pretty surprised and a little resistant to the notion of children with Jacobsen syndrome having autism. And then that's when we began to formally study it. And thanks to the people you've heard this morning, it's clear now that half of your children or so have autism. And then another tribute to the group was when Dawn was here with her family last time. That's when she told me about some cockamamie study going on by some wacko